Hey everyone, welcome back to another episode on genetics. Today what we're going to go over is a new chapter, chapter 5. And this chapter is mainly involved in gene linkage. So what gene linkage is involved is we're going to break apart the law of independent assortment right now, where sometimes some genes behave in a different way such that they're breaking the law of independent assortment, where you're getting gametes that appear at different frequencies for two or more genes, basically. And to introduce this topic to you guys, usually the people that were involved in this process were a couple of people. Baetson and Punnett, for example, they were involved, and Thomas Hunt Morgan. But basically what happened here was these three individuals, they took a couple of flies, or whatever it was. They took a couple of flies. Let's say they have a phenotype of we have the parent. They would be true breeding for purple, and they'll be true breeding for long, and we've crossed them with another parent that is true breeding for red and true breeding for round. And then what happens here, as we've come to expect from this cross, is that we have an heterozygous individual, and this individual will be purple and long. And then what we usually expect that what we're doing here, if we've crossed the F1 together again, what we should expect to see from a dihybrid cross is a 9:3 to 3:1 ratio. So let's write that down. So observed versus expected. So what we've expected to see in the F2, if we've crossed the F1 together, we expect to see a 9:3 to 3:1 ratio, where this is big A, oops, my bad, wrong letters, big P, big L, this is big P, little l, little p, big L, and then little p, little l, little l. So this is what we've expected to see, but however, what they found out was that you're getting a completely different ratio of what we're expecting in the F2 with Baetson and Punnett. So what they've looked at is that they found a completely different ratio of what we've expected to see. Baetson and Punnett found out that what's happening here in the F2 is that you're seeing a higher majority of individuals that have purple long phenotype and the red and round phenotype compared to what was expected. We've only expected 1/16 of individuals being red and round, but we're seeing a higher proportion of individuals that are red and round. Another thing that we're looking at too is that these individuals over here, we're seeing a lot lower proportion of these individuals for some reason, and they didn't really know. They had a couple of theories on this, but this is mostly involved in gene linkage. So basically there's a notion that you have a certain chromosome, and in this chromosome you have both of those genes that are close together. So let's say this is P and this is L, and then if they're very close together, they have a lower chance of recombining, and because they have a lower chance of recombining, you have a higher probability of developing a chromosome that has the exact same alleles, something like that, right? But whereas if they were really, really far apart between P and L, and if they were to undergo crossing over with another chromosome that is also very far apart, in this situation what happens is that because they're so far apart, that means that there's a higher chance of recombination happening. And then because of this, what happens is that your gametes are more likely to be equal in proportion. So yeah, basically that's basically what's happening over here. Just know that for now, that's all you pretty much need to know for this introduction episode. Another thing to look at is, we're covering over gene linkage right now, and we're seeing this primarily over here. But how can we actually determine from a data set, if there's gene linkage between two traits, something like this? And then you've probably already noticed the main thing that you should look for is that the parental phenotypes are higher in proportion than expected, and the recombinant phenotypes are less in proportion. So what's happening here is that our original parents were purple and long, and then we had another parent that was also red and round over here. And basically what we're saying over here from this is that our purple and long and our red and round original parents in the offspring appear at a higher frequency than the individuals that are recombinant, which are red long and purple and round. So basically this is a clue that you can use for a problem where you're given a data set and you're trying to figure out if there's gene linkage, and you're primarily using that assumption right here. So what you're doing over here is so we have a true breeding yellow tall plant, and we're crossing it with a red short plant. And then if we cross the F1 together, we recorded the data as yellow tall, red and short. And then if you read the problem carefully, we know that the parents were yellow and tall, and red and short. So since we're getting a higher proportion of parental phenotype over here compared to the recombinant phenotype, therefore these two traits are linked together through gene linkage. So hopefully that made sense. So yeah, that's all you have to do for if you're trying to figure out if there's if you have a data set and you're trying to figure out if there's gene linkage or not. Typically these problems you will be given an F1 that is pretty much all heterozygous most of the time, because you're crossing true breeding individuals together in the parents. So the F1 will always be heterozygous. And then because the F1 is always heterozygous, you should always expect a monohybrid dihybrid cross situation where you're getting a 9 to 3 to 3 to 1 or something like that. So because you're getting a 9 to 3 to 3 to 1, and there's a difference in this, then therefore it could be because of gene linkage, right? Another thing that you could also pay attention to is that sometimes in some problems you might be given gene linkage where the original parents were yellow and short, and they were red and tall. So therefore, if the original parents were yellow and short, we should see the increase in phenotype report, or increase the number of phenotypes for these particular individuals. But that's it for introduction to gene linkage. Next episode we're gonna go over how to from a data set how to figure out the recombination frequencies and stuff like that. So, but yeah, keep in mind that, and yeah, that's it. Have fun.